A point mutation is when one base is substituted for another. Missense, nonsense, silent, and read-through are all point mutations. These mutations can either be a transition or a transversion. Transition is when a purine is replaced with another purine, or when a pyrimidine with another pyrimidine, whereas transversion is when a pyrimidine replaces a purine, or a purine replaces a pyrimidine. Missense mutation is when a single base pair is changed and results in a different amino acid. Here, G has been changed to C. The substituted C has altered the codon sequence. Once translated, the third amino acid is now different. This mutation is a transversion because guanine, a purine, is being replaced by cytosine, a pyrimidine. Nonsense is a mutation that changes a codon into a stop codon. This makes the final protein strand shorter. Here, the A is substituted for T. This causes the resulting codon to change and code for a stop codon instead of lysine. This ends the protein strand early. This mutation is a transversion because adenine, a purine, is being replaced by thymine, a pyrimidine. A silent mutation is when a change in a base pair codes for the same amino acid. Here, C is being replaced by A, which causes the second codon to change from GUC to GUA. However, both GUC and GUA both code for the same amino acid. This mutation is transversion because cytosine, a pyrimidine, is being replaced by adenine, a purine. A read-through mutation is when a stop codon is changed to a normal codon, which makes the protein longer. Here, T is being replaced by C. This causes the stop codon, UAA, to change to a normal codon, CAA. This results in the amino acid continuing and making the protein strand longer. This mutation is a transition because thymine, a pyrimidine, is being replaced by cytosine, which is also a pyrimidine. Let's break down what we just learned with color beads and construction paper. These beads are going to be our bases, and the color blobs are going to be our amino acids. First, I will demonstrate missense mutation. 3 beads, red, blue, and blue, come together to form a group. This group of 3 mixes to create a purple blob. However, when one of the blue beads is changed to yellow, the beads all mix to create a different color blob that is brown instead of purple. Next, I will demonstrate nonsense mutation. Here we have a chain of color blobs. A particular color blob is made from 2 red beads and a blue bead. The blue bead is replaced with a red bead, which changes the color blob to all red, which turns it into a stop sign. This causes the color blob change to end after the stop sign, making the color change shorter than normal. Next, I will demonstrate a silent mutation. Here we have 2 blue beads and a red bead that combine to make a purple color blob. One of the blue beads is replaced with a purple bead. Even though the beads change sequence, the color blob remains the same. Next, I will demonstrate read-through mutation. Here we have a chain of color blobs with a stop sign being the last one. The stop sign is made from 3 red beads. One of the red beads is changed to yellow, which turns the red stop sign into an orange blob. Since there is no more stop sign stopping the color blob change, it continues and becomes longer than normal.